Hello students, I am R.K. Jha from Sri Chaitanya. Today I would like to discuss the topic microbes in human welfare. This topic is important as we are finding at least one question every year. Last year, that means in 2025, there were 3 questions asked. Before that, in 2024, only 1 question, and in 2023, no questions were asked. So let's see the topic. Microbes in human welfare. So if we take an average, it is 1 question per year. No question 2023. 2024 में 1 question asked. 2025 में you can see 3 questions. So these are the 2025 questions. Now this is the outline of the topic, that is microbe in human welfare. You can see microbes as a industrial products, microbes household products में, microbes in biofertilizers, microbes for the sewage treatment, microbes in the production of the biogas, and microbes for the as a biocontrol agents. Now this is the main topic. You can see microbes, they are present everywhere, that is they are ubiquitous. That means they are present in the soil, water, air, inside the bodies and that of the other animals and the plants. Even they are present in those habitats where other organisms generally don't live. For example, like thermal geysers, or in the layers of the thick snow, these microbes can be found. Microbes are diverse, such as protozoa, bacteria, fungi, and the microscopic animals and plant viruses, viroids, and the prions. They all come under the microbes. Prions are what? They are the infectious proteinaceous particle. These are the diagrams you can see. The most important, this is the diagram of a bacteriophage. This is the adenovirus, the same virus which is causing the respiratory infections. This is the TMV, tobacco mosaic virus. These are the colonies. You can see this is the fungal colony. This is what the bacterial colony. Microbes in the household products. So we know that the microbes, they are of course very harmful for us, but there are many microbes which is useful also. So let's see how they are useful to us. The first of all, microbes in household products. Here you can see that the lactobacillus and the lactic acid bacteria, they are used to convert the milk into the curd, and curd is more nutritious than the milk. Why? Because it is enriched with the vitamin B12. Another, we know that nowadays the very famous breakfast, idli and dosa, the batter is prepared through the fermentation process, and for that the certain bacteria they are needed, such as streptococcus is needed and the leuconostoc is needed. Now you can see कि for the bread making we are using the yeast Saccharomyces cerevisiae. In the same way, there are certain traditional drinks are also available which is also made through the fermentation process, and for that the yeast is used. Then certain foods, fermented foods are available, such as fermented fish, soybean and the bamboo shoot, and they are fermented through the microbes. Cheese, one of the oldest product of the microbe, that is very famous nowadays, and it is a very good type of food. Here the cheese, the different varieties of cheese are known by their characteristic texture, flavor and the taste. The specificity coming from the microbe used. For example, you can see the large holes in the Swiss cheese are due to the production of large amount of carbon dioxide by a bacterium called Propionibacterium shermani. The Roquefort cheese are ripened by growing a specific fungi, that is Penicillium roqueforti, on them, which gives them a particular flavor. The next is the microbes in the industrial product. In the industrial products are being formed using the big vessels called the fermenter, and the 2 main products are there. One is the alcohol, that is the beverages. The next is the antibiotics. Here the most important is कि the microbe which is used in the alcohol production is Saccharomyces cerevisiae, which is also called as the brewer's yeast. So here you can see कि they means this means fungi is used for fermenting the malted cereals and the fruit juices. Here, depending on the type of the raw material used for fermentation and the type of processing, such as with or without distillation, the different types of alcoholic drinks are obtained. Wine and beer are the product without distillation, whereas whiskey, brandy and rum, they are the product with distillation. Now come for the another, that is the antibiotics. Antibiotics, anti means against, biotic means life, against the life, but the against the life whom? That is the microbes which is causing the disease to us, and for us it is pro-life. The antibiotics are the chemical substances which are produced by some microbes and can kill or retard the growth of other microbes. And the first antibiotic discovered was, that is the penicillin, and this discovery is because of serendipity. What is serendipity? Discovery made by chance. Actually he was working on the bacterium Staphylococcus, and in that he was able to discover the antibiotic, that is the penicillin. The next, he discovered the antibiotic penicillin from the fungi, that Penicillium notatum. But you know, its full potential as an antibiotics was discovered or established by, that is the Ernest Chain and Howard Florey, and this was used during the World War II to cure the American soldiers. And nowadays also it is quite used because it is a very good type of antibiotic to cure several diseases. And nowadays the different type of antibiotics have been obtained to cure the diseases caused by different microbes. Now the next is, that is the chemicals, enzymes and other bioactive molecules. You know that the microbes are used to get the different type of chemicals, we are obtaining the enzymes, and also we are obtaining the different type of bioactive molecules which are useful to cure the different diseases. You can see Aspergillus niger, it is the source of citric acid, Acetobacter aceti, a source of acetic acid, Clostridium botulicum is a source of butyric acid, and the Lactobacillus is a source of lactic acid. The next is the yeast is used to make the alcohol, we already studied. Then the lipase is also obtained from the microbes such as yeast, say Candida, that is Candida lipolytica, say from it is obtained, and it is used in the detergent formulation and helpful in removing the oil stains from the laundry. Another is to clear the bottled juices. We also uses the two types of enzymes, that is the pectinase is used, another is the protease is used. Next, another enzyme, that is the streptokinase, it is used as a medicine to clear the blood clot from the blood vessels of the patients who have undergone the myocardial infarctions leading to the heart attack. You know, one important point I will tell you about this streptokinase, this is called tissue plasminogen activator. This is genetically engineered, you must remember that. Now the next bioactive molecule is the cyclosporin, and it is used as an immunosuppressive agent in the organ transplant patients produced by the fungus Trichoderma polysporum. The next is statin, which is used as a blood cholesterol lowering agents, and it is obtained from a yeast called the Monaskis purpureus. Next is microbes in sewage treatment. Municipal waste water is called the sewage, and the sewage has to be treated. Why? If it is not treated, then it will pollute our water bodies. The question comes ki how it is treated. The treatment of the sewage is done by the heterotrophic microbes present in the sewage naturally, and it is done in the 3 steps. That is, the first is primary treatment, second that is the secondary treatment, and the third, that is the tertiary treatment. Primary treatment is purely a physical process, it is purely a physical process, and in this the filtration and sedimentation is done. Secondary treatment is purely a biological process in which the organic matter present in the sewage is degraded using the aerobic microbes such as bacteria and the fungi. They forms the Flocs. Now here one important point is that is the biological oxygen demand, or that is the BOD. What is BOD? It is the amount of oxygen required for the degradation of all the organic matter present in 1 liter of the sewage through the oxidative process by the microbes. More the bod of a water, that means more polluted that water is there. Next is after the secondary treatment, means in the secondary treatment, once the bod has reduced to the considerable level, after that it is sent to the settling tank where all the Flocs they get settled at the bottom, and the small portion of that it is transferred as a inoculum. What do you call that? The settled Flocs. They are called the activated sludge. And after removal of the small amount and it is putting back into the aeration tank, rest all are transferred into the anaerobic sludge digester where it is digested anaerobically using the methanogens, and it produces the biogas. Even this also comes under the secondary treatment only. Now after secondary treatment, the water is discharged into the water body. Now if the same water has to be used for drinking or the household purposes, then we have to go for the tertiary treatment. That is, the tertiary treatment is a physico-chemical process in which we have to go for the ozone treatment, chlorine treatment, or even we have to go for the RO treatment. Now the Ministry of Environment and Forest has initiated the Ganga Action Plan and the Yamuna Action Plan to save the major rivers of the country such as Ganga and the Yamuna. The next is microbes in the production of biogas. First of all, we have to know the biogas consists of what kind of gases. It consists of methane, that is 50 to 70%. Then it is the carbon dioxide, which is 20 to 30%, and in remaining the other gases like H2, H2S, etc. are there, and these are produced by the action of the methanogens, that is the Methanobacterium, and this production is occurring under the anaerobic condition, that is the fermentation process is involved here. Cellulose, which is the major component, they are digested through this process. You know, these methanogens are also present in the rumen of the cattle. Why? Because the cattle eat the plant parts, and plant parts they are having the cellulose. So cellulose has to be digested. That's why the methanogens are present in their rumen. You can see this biogas plant is there. This plant is established under the guidance or efforts of Indian Agricultural Research Institute and the Khadi and Village Industries Commission. You can see this is the biogas plant. Here this is the digester, gas holder is there, and this is from this side dung and the water is, that is the slurry is fed, and after this is the gas holder, and this is from where the gas is, this is connected with the pipe and the gas moves out from here, and finally the sludge can be taken out from this side. Now next is the microbes as the biocontrol agents. So biocontrol refers to the use of biological methods for controlling the plant diseases and the pest. In modern societies, these problems have been tackled increasingly by the use of chemicals, that is by the use of insecticides and pesticides. These chemicals are toxic and extremely harmful to human beings and the animals like and have been polluting our the environment like soil, groundwater, fruits, vegetable, crop plants, etc. Now biological control of the pest and diseases, it is basically based on the natural predation rather than that is introduce the chemicals. Here a key belief of the organic farmer is the biodiversity furthers the health. The more diversity a landscape has, the more sustainable it is. So organic farmer therefore works to create a system where the insects that are sometimes called the pests are not eradicated completely, rather they are controlled. So here They are kept as the manageable level by a complex system of checks and balances within a living and the vibrant ecosystem. Now you can see here an important part of this biological farming approach is to become familiar with the various life forms that inhabit the field, that is the predators as well as the pests, and also their life cycles, pattern of feeding and the habitats. Because of that only we will be able to establish the food web, and that this will help develop the appropriate means of biocontrol. Now you can see there are certain the animals such as the ladybird and dragonflies are there. They are used to get rid of the aphids and the mosquitoes. Now if you want to kill the butterfly at the caterpillar stage, then we can use the Bacillus thuringiensis, and you know its spores are available in the sachets. The spores are mixed with the water and it is sprayed over the plants like brassicas and all. When it is eaten by this butterfly caterpillar, it goes inside the gut where the toxins gets activated, and thus the caterpillar they are getting killed. Nowadays through genetic engineering we can take out the genes responsible for the production of that toxin, and we can insert the plant and directly we can make the plant resistant to these insect pests. The another is biocontrol agent mein we are using the trichoderma. Trichoderma is basically a soil borne free living fungi is there, and it protects the root ecosystem. They are effective biocontrol agents of several plant pathogens. The next is Baculovirus, are pathogens that attack the insects and the other arthropods. The majority of this Baculovirus used as a biological control agents are in the genus Nucleopolyhedrovirus. These viruses are excellent candidate for the species specific narrow spectrum insecticidal applications. They have been shown to have no negative impact on the plants, mammals, birds, fish, or even non-targeting insects. So this is especially desirable when the beneficial insects are being conserved to aid in an overall the integrated pest management program, or when an ecologically sensitive area is being treated. The next is microbes as the biofertilizers. There are various bacteria, fungi and cyanobacteria, they are used as biofertilizers. You know, bacteria mein rhizobium are used as a symbiotic nitrogen fixer. It is symbiotic with the root nodules of leguminous plants. Then there are certain bacteria which can fix the nitrogen in the free living conditions also. For example, that is the Azospirillum and the Azotobacter. Now there are certain fungi also used as a biofertilizer, such as they are called the Mycorrhiza. The example here you can see the genus, fungal genus, that is the Glomus. It is making an association with the roots of the orchids, and then it is absorbing the phosphorus, mainly the phosphorus for the plant. But you must remember one thing, Mycorrhiza never fix the nitrogen. This is very important. The next is कि Mycorrhiza, it's not only providing the phosphorus to the plants, it's also protect the root ecosystem from the soil borne or root borne pathogens. It also makes the plant tolerate to the salinity as well as tolerating it to the droughts, and an overall increase in the plant growth and the development. The next is that is the Cyanobacteria. Cyanobacteria, they are autotrophic nitrogen fixers, and the most common nitrogen fixers among the Cyanobacteria, they are Anabaena, Nostoc and the Oscillatoria. They are fixing the nitrogen in the paddy fields, especially the Oscillatoria is one of the potent nitrogen fixer in the paddy fields. Even the Anabaena is also, Anabaena and Nostoc both can fix the nitrogen in both symbiotic condition also and in the free living condition also. Oscillatoria can fix the nitrogen only in the free living condition. So this is all about the topic we have seen. Now we can see There are certain important things, that is the summary, not to be missed. Very important. Questions are framed from here. This is the summary part, you can see. Next, the important diagrams: bacteriophage, adenovirus, TMV. These are the colonies. This is the bacterial colony, this is the fungal colony. You can be given the question to identify. This is the biogas plant, you can see, and labeling can also be given in the exam. You can be given to identify what exactly is this, as well as you have to be given to label it also. Next, these are the important points you must remember, like all the microbes, what they are exactly beneficial to us, they have been compiled here. This is biogas, you can see methane 50 to 70%, carbon dioxide 30 to 40%, and in traces में H2, N2, H2S they are present. Microbes, methanogens, bacteria, methanobacterium, technique developed by Indian Agricultural Research Institute and the Khadi and Village Industries Commission. Microbes as a biocontrol agent, biofertilizers, you can see all this. Now there are certain important terms: sewage, primary treatment of sewage, primary sludge, Flocs, secondary treatment, biological oxygen demand, activated sludge, and these are the questions that you have to do it. They are the important type of questions. You should not miss it.